The dates is Professor Will, CBMD, PhD, today Saturday, December 22nd, 2018 at 16:04, 24 seconds, Eastern Standard Time, Science News. Baby star's fiery tantrum could create the building blocks of planets. Date December 21st, 2018. Source University of Warwick. Summary: a massive stellar flare on a baby star has been spotted by astronomers shedding light on the origins of potentially habitable exoplanets. One of the largest ever seen on a star of its type. The huge explosion of energy and plasma is around 10,000 times bigger than the largest solar flare ever recorded from our own sun. The discovery is detailed in a paper for the monthly notices of the Royal Astronomical Society and reveals how this huge tantrum could even perturb the material orbiting a star, which would create the building blocks for future planets. The flare was seen on a young M-type star named NGTS J121939.5 355557, located 685 light-years away. At around 2 million years old, it is what astronomers refer to as a pre-main sequence star, which is yet to reach the size that it spends the majority of its life cycle. It was observed as part of the large flare survey of thousands of stars by University of Warwick, searching for explosive phenomena on stars outside our solar system. Researchers used the Warwick LED Next Generation Transit Survey NGTS telescope array in Chile, which is designed to find exoplanets by collecting brightness measurements of hundreds of thousands of stars, and is based at the European Southern Observatory Panel Observatory. Research attention was drawn to NGTS J121939.5-35557 as it had one of the largest flares seen in these types of stars. A stellar flare occurs when the magnetic field of a star rearranges itself, releasing huge amounts of energy in the process. This accelerates charged particles or plasma within the star, which crash into its surface, heating up to around 10,000 degrees. That energy produces optical and infrared light, but also X-rays and gamma rays that can be picked up by telescopes on Earth and in orbit. Magnetic fields on M stars are a lot stronger than those on our own sun, and the astronomers calculate that this size of flare is a rare event, occurring anywhere from every 3 years to twice a decade. Then on this one particular night, we saw it suddenly grow 7 times brighter than normal for a few hours, which is pretty extreme, and then after that it goes back to normal. We see these types of flares on the sun, but nowhere near as big as this. On our sun, you can do incredibly detailed studies on this kind of activity. It is difficult to extend that understanding to other stars because the data we need hasn't been available until now. This is an incredibly young star, only about 2 million years old. You'd call it a baby. It is going to live for 10 of millions of years, so it is in the first 1% of its lifetime. Even though it is much cooler than our sun, by about 2,000 degrees, it is roughly the same size, but pretty large for an M star. This is because it is still being formed from gas in the disk, and contracting and cooling until it reaches the main sequence, staying at a certain radius and luminosity for billions of years. Finding out these kinds of details has only been possible thanks to the Gaia mission that began earlier this year. X-rays from these large flare events are thought to affect the formation of chondrules, flash-melted calcium aluminum-rich grains, in the solar's protoplanetary disk. These gather together into asteroids that eventually coalesce into orbiting planets. The study adds to understanding how flares perturb the protoplanetary disk, moving around the material that impacts on planet formation and affecting the eventual structure of a planetary system. A massive flare like this could be advantageous for planet formation, or it could be disruptive. This particular star won't have formed its planets yet, so this type of flare activity is something that astronomers will need to take into account when considering planet formation. There's a discussion at the moment around whether flares are a good or a bad thing for life on orbiting habitable planets, because they output a large amount of UV radiation. That could cause biological damage to surface organisms and damaging their DNA. On the other hand, UV radiation is required for various chemical reactions to start life, and that's not typically provided in great enough quantity by these types of stars. These flares could potentially kickstart these reactions. Today's is Professor Will, CBMD, PhD, today Saturday, December 22nd, 2018, at 17:24, 35 seconds Eastern Standard Time. Himalayan marmot genome offers studies to life at extremely high altitudes. Date December 20, 2018. Source Cell Press. Summary: Himalayan marmots can survive at altitudes up to 5,000 meters in the Himalayan regions of India, Nepal, and Pakistan, and on the Qinghai, Tibetan Plateau of China, where many of them face extreme cold, little oxygen, and few other resources. Now researchers have sequenced the first complete Himalayan marmot genome, which may help them to better explain how the marmots live in such extremes. The findings, which appeared December 20th in the Journal of Science, hint at genetic mechanisms underlying high-altitude adaptation and hibernation, the researchers say. They also serve as valuable resources for researchers studying marmot evolution, highland disease, and cold adaptation. As one of the highest altitude dwelling mammals, the Himalayan marmot is chronically exposed to cold temperature, hypoxia, and intense UV radiation. They also hibernate for more than 6 months during the wintertime. Researchers consider the Himalayan marmot as an ideal animal model for studying the molecular mechanisms of adaptation to extreme environments. To begin, they sequenced and assembled a complete draft genome of a male Himalayan marmot. They also resequenced 20 Himalayan marmots, including individuals living at high and low altitudes, and 4 other marmot species. Additionally, RNA sequencing was done to compare gene expression differences between marmots in a state of torpor and awake marmots. The DNA data show that the Himalayan marmot diverged from the Mongolian marmot about 2 million years ago. The researchers identified 2 genes, SLC25A14 and AMP, a process pseudogene that have been selected in different directions in mammals living at low versus high altitudes, suggesting they are related to survival in high-altitude populations under conditions of extreme low oxygen. They further suggest that SLC25A14 may have an important neuroprotective role. The shift in question, AMP affects the stability of RNA encoding gene. AMP, which may be a protective strategy to prevent the excess growth of new blood vessels under extremely low oxygen conditions. The RNA sequencing data show that gene expression changes occur in the liver and brain during hibernation. These include genes in the fatty acid metabolism pathway as well as blood clotting and stem cell differentiation. Interestingly, a previous study suggested that because the hibernator's brain is exposed to near freezing temperature and has decreased blood flow, there is an increased risk of blood clots, the researchers note. Their brain stem cells may also be better prepared to repair injuries as an adaptation needed to survive extreme environmental stresses. The researchers say they plan to continue improving the quality of Himalayan marmot's genome. They note that the Himalayan marmot is also known for being highly susceptible to woodchuck hepatitis virus and is a natural host and transmitter of the plague to humans. We will elucidate the immune system feature responsible for the hepatitis virus and bacterial infection. Today's is Professor Will, CBMD, PhD. Today, Saturday, December 22nd, 2018 is 17:40, 27 seconds Eastern Standard Time. Science News. Small changes in oxygen levels have big implications for ocean life. Date December 19, 2018. Source University of Rhode Island. Summary. Oceanographers have found that even slight levels of ocean oxygen loss or deoxygenation have big consequences for tiny marine organisms called zooplankton. Zooplankton are important components of the food web in the expanse of deep open ocean called the midwater. Within the slice of ocean below the surface and above the seawater are oxygen minimum zones, OMZs, large regions of very low oxygen. Unlike coastal dead zones, where oxygen levels can suddenly plummet and kill marine life not acclimated to the condition, zooplankton and OMZs are specifically adapted to live where other organisms, especially predators, cannot. But OMZs are expanding due to climate change, and even slight changes to the low oxygen levels can push zooplankton beyond their extraordinary physiological limits. All the animals in the ocean's oxygen minimum zone have adapted over millions of years to the very low oxygen of this extreme widespread midwater habitat. They are living at the very limits of their physiological capability. Our research shows that they are sensitive to very small changes in oxygen and decrease in abundance when oxygen gets just a little bit lower. The research team, which this week published their findings in Science Advances, found more natural variability in oxygen levels in the OMZ than previously known. This has a direct effect on the distribution of many types of zooplankton because, as the team discovered, the organisms respond to a less than 1% reduction in oxygen levels. While zooplankton have made millions of years to adapt to conditions in the OMZ, these low oxygen zones may expand rapidly due to climate change, leading to major unanticipated changes to midwater ecosystems. For example, an expansion of the OMZ into shallow waters may make zooplankton more susceptible to predators like fish. If this leads to a zooplankton population crash, it will have impacts all the way up to the food chain. Further loss of oxygen in ocean waters is predicted in the future as a result of global warming, and these animals may be unable to adapt and persist. They are important components of the food web, oceanic ecosystems, and their loss could potentially impact top predators, including whales and commercially important fisheries. Today's is Professor Will, CBMD, PhD. Today, Saturday, December 22nd, 2018 is 18:09, 24 seconds Eastern Standard Time. Science News. Biologists turn eavesdropping virus into bacterial assassins. How cross-kingdom communication led to a breakthrough phage therapy. Date December 13, 2018. Source Princeton University. Summary. Researchers have found a bacteria-killing virus that can listen in on bacteria conversations, and then they made it attack disease including Salmonella, E. coli, and cholera. The idea that a virus is detecting a molecule that bacteria use for communication that is brand new. Researchers found this first naturally occurring case, and then they rearranged that virus so that they can provide any sensory input they choose, rather than the communication molecule, and then the virus kills on demand. A virus can only ever make one decision: stay in the host or kill the host. That is, either remain under the radar inside its host or activate the kill sequence that creates hundreds of thousands of offspring that burst out, killing the current host and launching themselves toward new hosts. There is an inherent risk in choosing the kill option. If there are no other hosts nearby, then the virus and all its kin just died. VP882 has found a way to take the risk out of the decision. It listens for the bacteria to announce that they are in the crowd, upping the chances that when the virus kills, the released viruses immediately encounter new hosts. It is brilliant and insidious. This paper provides an entirely new perspective on the dynamic relationship between viruses and their hosts. This study tells us for the first time that when a phage is in its lysogenic state, it is not fast asleep, but rather catnapping with one eye open and ears alert, ready to respond when it hears signals that cells are getting ready to respond to changes in their environment. Researchers had discovered years before that bacteria can communicate and sense one another's presence, and that they wait to establish a quorum before they act in concert. But she had never imagined that a virus could eavesdrop on this quorum-sensing communication. Researchers' work shows that these quorum-sensing molecules are conveying information across kingdom boundaries. Viruses are not in the same kingdom as bacteria. In fact, they are not in any kingdom because they are not technically alike. But for such radically different organisms to be able to detect and intercept each other's signals is simply mind-boggling. It is not like enemy nations spying on each other, or even like human communicating with a dog. Those at least are members of the same kingdom, animal and phylum vertebrate. The idea that there's only one example of this cross-domain communication made no sense to us. Researchers discovered the first case, and then they discovered in hand. They went looking more deeply and they found a whole set of viruses that harbor similar capabilities. They may not all be listening to the quorum sensing information, but it is clear that these viruses can listen in to their host's information and then use that information to kill them. Communication seems like such an evolved trait. To hear that bacteria can do it. Their discovery, it was just mind-blowing that organisms you think of as so primitive could actually be capable of communication. And viruses are even simpler than bacteria. The one I studied, for example, only has about 70 genes. It is really remarkable that it devotes one of those genes to quorum sensing. Communication is clearly not something higher organisms created. Once researchers demonstrated that VP882 was eavesdropping, it began experimenting with feeding it misinformation to trick the virus into killing on command, to turn the predator into an assassin. VP882 is not the first virus used as an antimicrobial treatment. Viruses that prey on bacteria called phages, and phage therapy targeting a bacterial disease with a phage is known as medical strategy. But VP882 is the first phage that uses eavesdropping to know when it is optimal to kill its targets, making experiments with Salmonella and other disease, causing bacteria the first time that phage therapy has used trans-kingdom communication. In addition, this virus holds enormous promise as a therapeutical tool because it does not act like a typical virus. Most viruses can only infect a very specific type of cell. Flu viruses, for example, only infect lung cells. HIV only targets specific immune system cells. But the virus VP882 has an exceptionally broad host range. So far, research has only performed proof of principle tests with 3 unrelated bacteria: Vibrio, cholera, Salmonella, and E. coli. Those diseases have evolved separately for hundreds of millions of years, so the fact that they are all susceptible to this bacterial assassin suggests that many more are as well. Antibiotic resistance is clearly a major global health threat, and there is a clear and evident demand for new strategies and approaches to this problem. Although we have admittedly found it tricky even to reach first base with basic therapeutical use of naturally occurring phages, we can envisage the possibility of a home run if we can engineer phages for therapeutical use that have very specific targeting. These viral assassins might even slow down the emergence of antibiotic-resistant strains. Today's is Professor Will, CBMD, PhD. Today, Saturday, December 22nd, 2018, is 18:51, 20 seconds Eastern Standard Time. Science News. Brainwaves suppress obvious ideas to help us think more creatively. Date December 10, 2018. Source Queen Mary, University of London. Summary. The human brain needs to suppress obvious ideas in order to reach the most creative ones, according to scientists. These obvious associations are present in both convergent thinking, finding an out-of-the-box solution, and also in divergent thinking when individuals have to come up with several creative ideas. Creativity requires us to break away from more common and easily reached ideas, but we know little about how this happens in our brain. A new study published in the Proceedings of the National Academy of Sciences shows that brainwaves play a crucial role in inhibiting habitable thinking modes to pave the way to access more remote ideas. The researchers found that these brainwaves, or alpha oscillations, in the right temporal area of the brain increase when individuals need to suppress misleading associations in creative tasks. These obvious associations are present in both convergent thinking and also in divergent thinking when individuals have to come up with several creative ideas. The researchers show that stimulating the right temporal part of the brain in the alpha frequency increases the capability of inhibiting obvious things in both types of creative thinking. This was demonstrated by applying an electrical current to the brain through a non-invasive technique called transcranial alternating current brain stimulation, TACS, which causes minimal to no side effects or sensations. The findings have implications for how we understand creativity and opens up potential ways of affecting the creative process, including by using TACS. If we need to generate alternative uses of a glass, we first must inhibit our past experience, which leads us to think of a glass as a container. Our study's novelty is to demonstrate that right temporal alpha oscillations is a key neural mechanism for overriding these obvious oscillations. In order to understand the processes underlying the production of novel and adequate ideas, we need to break down its constituent processes, dissecting creativity as much as possible at first, and then analyzing them in context, before putting them back together to understand the process as a whole. The researchers demonstrated the neural mechanism responsible for creativity by monitoring the brain's electrical activity through electroencephalogram, EEG, which picks up electrical signals through small sensors placed on the head. Using TACS also enabled them to probe the wave's causal role. The experiments they conducted looked at how the brain tackles a series of creative tasks, such as finding words that link to one another. For example, every time we search for concepts associated with a word, we start from stronger associations and move progressively toward weaker or more remote ones, e.g. cat, dog, animal, pet, human, people, family. Previous studies show that some people are more creative than others because they are able to avoid strong associations in order to reach more remote ones, and this study demonstrates that the alpha brainwaves are crucially involved in this process. The researchers hope to understand how neural processes are integrated when solving creative problems out of the laboratory setting, and whether it is possible to build stimulation devices which can monitor the brain and stimulate creativity whenever needed.